switch to presenting my my previous work, my background, and I'm just gonna brush over a few studies, and then I will talk about my project for my lab here at Penn State. I'm going to go very quickly about my background, but I thought it was going to be interesting that you know that I'm not always been a soil scientist. In fact, now that I think about it, because I'm talking right after Sharifa, I probably should have included that before my PhD, I did a master in plant pathology, and in fact, I worked on Colicotrichum lindemisianum. So we have more in common than I thought. My PhD was in Paris, and I worked at the interface between geoscience and microbial ecology. I was co-advised. I worked primarily on how microbes precipitate mineral carbonates in the form of rock, so microbialites, but also I worked on how some cyanobacteria can control the mineralization of carbonate, and I discovered that some of them are able to precipitate these beads inside their cell, which was very new at the time in geomicro. And I worked more in this kind of topic as a postdoc, where I was working on how cyanobacteria dissolve mineral carbonate, which is a geochemical paradox because you have to couple oxygenic photosynthesis to the dissolution of mineral carbonate, and those play in the opposite way on the carbonate balance. Because photosynthesis tends to increase the pH, and you need to decrease the pH if you want to dissolve carbonate. I don't know what it's, yeah, it's moving by itself, I'm sorry. And during a first postdoc in Arizona, I discovered soil, and I saw that, you know, this was the area where I wanted to go, because working on soil, you can really have questions that have a global impact. So I worked on biological soil crust. It's a very specific type of soil from Ireland, and I'm gonna talk to you a little bit more about them. And I worked again with them. So I used different methods from molecular ecology to different type of imagery to try to understand them better and to understand the microbe in the context of these soils. And more recently, I was also in California at the Joint Genome Institute, which is in the Lawrence Berkeley National Lab. And I worked on developing a method to probe active microbe in soils, which is called Bondcat. And again, I'm gonna talk to you a little bit more about this one. Something that I wanted to mention to foster collaboration, just a summary of the thing that I'm interested in, like very generally, soil and molecular ecology and microbiome are probably my primary interest, but I also have a background in biogeochemistry, mineralogy. And so because I worked in different fields, I kind of have accumulated different set of skills. So some that are very common among this group of people in molecular ecology or microbiology, but maybe some other that are not so common in imagery and spectroscopy, including stable isotope and synchrotron-based techniques. So I'd be happy to talk with you about how we can implement these things in your research if you are interested. Okay, so let's talk about what I want to focus on, my research moving forward. So as you may know, soils are really key to answer some of the global challenges that we are facing, including climate change, crop yield and food production, and also soil degradation and pollution. And if we look at this big like theme of research, there is something that I think is a common denominator, and that's microbes. Because when you think about climate change, almost all the organic matter that is in the soil is of microbial origin or was turned out by microbe very recently. And so, you know, like to mitigate the amount of carbon that we have in the atmosphere, we could play on this giant pool of carbon, which is more than 2 times, in fact, that is in the atmosphere. There is also a dialogue between microbe and plant in the rhizosphere, so we could use microbe as natural fertilizer to decrease other type of chemical fertilizer. And soils are a very big biodiversity reservoir with a lot of natural products to be discovered. And there's also potential for microbial restoration and remediation of soil pollutants. So those are some of the big questions that I think you know, I would like to keep in mind while working in soil in my lab. One of the things about soil that make it difficult to work, and it's like everybody who's worked with soil know that it's quite difficult to as a system because it's very diverse. So in each gram of soil you have billions of cells and tens of thousands of species usually. And together these species, they perform globally relevant processes in a context that, you know, that is very complex because you have the soil matrix with all the mineral, all the organics, you have a fluid, and you have also an atmosphere in the soil. And so it's very difficult to understand how these things work together at this very small scale. One of the ideas that is around in the literature is in fact that beyond the hotspots that are, for instance, the rhizosphere or close to the earthworms, there is not much happening. So soils are microbial desert populated by scattered inactive microbes. So we could think like that anything that is away from a hotspot is not relevant. But in fact, you know, there are still billions of cells there. And we know that, you know, what is in the rhizosphere cannot account for everything that is turned over by the soil. So they have to be active and they in fact have to process carbon, nitrogen, and like all the elements in the soil. On top of this kind of complication due to the biology of the system, we also have some technical issues in soil because we know that up to 40% of the DNA that we retrieve from soil comes from free DNA or from cells that are physiology impaired. So when you think about relating the microbial diversity to the soil physiology, this is really an issue because up to 40% of the data that you retrieve are noise to try to link these two things. On top of that, we know that a lot of cells can be dormant. So they are estimated all over the place from like 1% to 99%. But that's something that we also need to clarify in order to understand, you know, which microbes contribute to which function in the soil and when. So I'm going to talk to you quickly about two projects that I've been working on trying to address this question. The first one is going to be about, it's more like a kind of on the side of method development and what we can do to address this question better. And the second one is more scientific example on how we can use this type of thinking on how to relate microbial diversity to soil properties in an ecological context. So first of all, I wanted to talk to you about this new method called Bondcat. So the idea of that project was to link better who is there and what they do and how they do it. So one step you can take to bridge these two questions is to filter who is there by who is active. So you only look at a sub-selected group of cells that are the active one and that in fact contribute to the activity and the physiology of what is happening in the soil at a larger scale. To do that, I, for the first time, transferred a technique that was first developed for mammalian cells and then used in marine sediment to the study of soil. So it's called bio-orthogonal non-canonical amino acid tagging. That's a very long name for something that is in fact really simple. So it's a 3-step process where you incubate the soil with a modified methionine. So you can see this methionine, I don't know if you can see my mouse maybe. This methionine has an additional alkyne group here. And so it will be incorporated into proteins, and we actually check that, you know, you have to have active cells in the soil so that it happens. It cannot just diffuse into a cell. It has to be incorporated into proteins. And then after the incubation, you can get the cell off the soil, which is another question that I'm happy to discuss how you do that and how efficient that is. So when you get the cell off the soil, you can then further click So it's the click means it's the cycloaddition of azide alkyne copper catalyzed cycloaddition of these two groups here that form this triazyl conjugate. So you link the newly made protein to a fluorescent dye in that case. And then in that project, I use fluorescence activated cell sorting to sort, to physically separate the cells that were fluorescent so that were making protein during the incubation from the other ones. And so to make this kind of long story short, what I did after that is that I separated cells that were active from non-active. I constructed 16S library on and did amplicon sequencing on the cell pellet. And I'm just going to show you one graph, which is an ordination of all my libraries from this project. And just the thing that you have to see is that all my sample, my control, and the inactive samples kind of fell together here. And so this is an ordination of the Berry-Curtis pairwise distances. Sorry, I should have said that before. And the active one actually fell together, and that was significant. And so I could go into more detail, you know, just dissecting who was active when they were active, but that graph by itself really validated the use of Bondcat in soil, because our data were reproducible, you know, like all the samples coming from the same soil at the same time point were together, samples coming from the same depth were also together, and they were different from the inactive fraction. So we thought that was pretty exciting, because it's usually hard to get something reproducible in soil when you start from a different gram of soil, just because you have so much diversity. But we think it will have a lot of value in studying soil communities to just focus on active cells. And so I can comment on that more if you're interested, but if you compare this technique to, for instance, stable isotope probing, it also has a lot of value, because you can have a smaller, a better snapshot of what is happening at smaller time scale, and it's also way less intensive in terms of bench work. So to conclude on that part, what we found here is that there is this traditional view that most microbes might be inactive beyond the hotspot in the soil. In fact, in our study, we could see, you know, the amount of microbes that turn active, and we found up to 70% in our soil were active. So it gives a new perspective on, you know, what can be happening in soils, and maybe those cold spots are not so cold. With this method, you can imagine that you can vary the incubation condition to see if you wake up different microbes. You can think about looking in rhizosphere microbe versus soil microbe. So there are lots of opportunity to expand on using that technique. That's why I wanted to mention it. And now I'm going to talk about another study where, again, around the idea of linking microbial diversity to soil emergent properties, I worked on how microbe control soil albedo. So the albedo, in case you are not familiar with it, is a measurement of how much of the total irradiance that comes to a surface is absorbed versus reflected. So if you have a low albedo, like when you wear a black t-shirt, there a lot of the incoming irradiance and energy is absorbed by the surface, and a little bit is reflected. So typically a low albedo surface will have a warmer temperature than a high albedo, like the snow, for instance, that reflects a lot of energy back. So for this study, I worked with my favorite model system, which are biological cyclists. So I like them because there are no plants there, and plants are complicated. So these soils are a completely microbial system. Right now, they cover 12% of Earth's continental area, and they are very major in the fixation of nitrogen and carbon in land. So those numbers, I'm not completely sure I trust them. They are from a paper, but I mean, even without considering those numbers, even if it's a few percent, it's still like a large fraction that we need to consider. And we also know that they are endangered by climate change. There have been some models showing that 40% could disappear within the next 30 years. They are very important for ecological services in the area where they are, because they are the first community that populate bare sand, usually bare ground, and they are responsible for soil stability and soil fertility in arid land. So when you look inside biocrust, it's actually pretty cool. So this is like a cross section. So this is the top of the soil, and this is inside the soil. So in gray are the soil particles, and in red it's an overlay of the autofluorescence of cyanobacteria. Because in fact there is no plant, but you still need to input some carbon in there, because there is no carbon and no nitrogen in the soil when you start. So the carbon is fixed by cyanobacteria that kind of play the role of a leaf and a root, because they form these big bundles. So you have here a bundle under a light microscope. So you see like it's multiple filaments, sorry, it keeps moving, within a sheath. So this is like a cross section. So each filament, they share a common sheath, and then they stick the soil grain together. Obviously here you only see the autofluorescence of cyanobacteria, but this community are way more diverse. We know that the cyanobacteria come along with some nitrogen fixers in their sheath, like a little community of pioneers. And then you find a lot of other species that are in the soils that mostly rely on the exudates, or the carbon that is deposited by the cyanobacteria for living. These systems are also very cool to work in the lab, because you can sample them when they are dry, and all the biomass is really within the first millimeter. So for a soil, you don't have to deal with a different type of horizons. You have all the community in the top millimeters. And when you call them, and then in the field, they are dry, so they are inactive, and you can store them for years. And then when you wet them, simulating a rain event, they turn active again. So they are a very cool model system for soil to work in the lab. So the particular study I'm going to talk about is originated from another study that was published in 2013, where my former group looked at so this cyanobacterium, which I didn't say, it's called microcolis. And they looked at the distribution of microcolis at the continental scale in US. So you can see this is California and Arizona and New Mexico. And they found that there are two species of microcolis, Microcolis vaginatus and Microcolis trintupiae. And they found that there was a biogeography at the continental scale. Like Microcolis trintupiae liked hot desert, while Microcolis vaginatus liked cooler desert. And that was recapitulated in culture. So when you cultivate them, you see that Microcolis vaginatus grows better at like 15 Celsius, while Trintupiae grows better at more like 25, 28 Celsius. That was the first time that, there was an example of biogeography for microbe, and also prediction on, as climate change happen in this region, you expect vaginatus to disappear and be replaced by Trintupiae. So they are still monitoring this region to see if this is in fact happening. But that was the idea that started this project, because then we were thinking about what can drive this shift of microbe that sometimes happen at a smaller scale. So you still find both species everywhere. It's just one dominates over the other. So I worked on a set of samples that came from a very small site. So they were all sampled within 2 meters. And first I measured the albedo. So, sorry, we know that in the field you can find crust of different colors, and the colors is linked to the level of maturity of the crust. So as the crust get more and more mature, they are darker and darker. And so in that case, I measured the albedo, and I found what I was expecting, which is that, darker crust have a lower albedo than the light crust. And they behave the way that you expect in terms of temperature. So I put them on the roof of the building I was working in Arizona. And so you see this is Celsius. So it went up to more than 55 Celsius that day in Arizona. And I went there and I measured the surface temperature of these samples during the day. And so you can see that, you know, the lighter samples had lower temperature than the darker sample as expected. But what we did not really expect was that sometimes the difference was as much as 10 degrees Celsius, which is a huge difference when you think about biological activity. Because like for some of these samples in the field, it would be the difference between being frozen and being non-frozen, so potentially being active altogether. So then we thought about how to explain that difference, and we know that the color of the crust is due to pigments. So I extracted full pigments from this crust, and here you have the absorbance of these pigments, and this is the wavelength, so it's just like a regular visible spectrometry in the visible, sorry. And so the dotted line is the full spectrum of the total crust pigment, and I overlaid the absorbance of two pigments that we know are there. One is cytonemine, and the other one is chlorophyll A. So you see that, in fact, cytonemine is a major pigment that absorbs a lot here in the UVA, and then a little bit everywhere, in fact, in the visible. And we know that this pigment is only produced by some cyanobacteria from the order of the Nostocales, and this is how they look in culture. So they have this brown color because they produce this pigment. And we know that with maturity, they can colonize the surface of the crust and produce this pigment that might be responsible of the change of color. And in fact, when we try to correlate the albedo with the concentration of cytonemine, we find that there is a good correlation. So we hypothesize that it's a colonization by these microbes, these cyanobacteria that produce this pigment, that are responsible to the change of color and the change of albedo of this soil. And more than that, we in fact found out, which is pretty common, that the albedo directly relates to the surface temperature in these soils. So we hypothesize that it's the microbes that are colonizing this soil that are changing the soil surface color by producing a specific pigment that change the albedo, and that also control, in that case, the soil surface temperature. Just so you know, cytonemine is a very cool pigment because it's absorbed in the UVA, so it takes away this highly energetic photon that could hurt the photosystem, for instance, but then it doesn't emit any light in the visible. Everything is emitted in the infrared, so it is in the form of heat. So it also makes sense that you know, it correlates with the soil temperature. And of course, we were interested to look at how that affected the community that live inside, so below the surface of the soil. So these are the results just for the cyanobacteria. And here you are looking at a plot that shows you the amplicon sequencing breakdown of cyanobacteria in the different sample. So you have like 6 replicates per sample, and sample 1 is the lightest sample, and then it goes to the darkest sample is sample 5. And what you can see, so first of all, you can see that there are more of the Nostocales which produce cytonemine, so those are the red thing in the darker crust. But also something that was very striking was that we saw a replacement of Microcoleus vaginatus, which, if you remember, is the one that like cold climate, by Microcoleus trintupiae as the main architect of this crust, which is the one that like more hot desert. So we saw at a small scale something that recapitulates the continental scale finding that was published before, and we thought that was very cool because that means that, you know, even at this scale, the temperature is a very good predictor of which is the main cyanobacterium that is the architect of the community. So to summarize this part, we saw that you know, the deposition of cytonemine can affect the soil surface color and soil surface albedo, and we hypothesize that this is these microbes that produce cytoamine that are responsible for this change of albedo that in fact can change the soil temperature up to 10 degrees Celsius and has consequences on the underground microbial community with a replacement of the keystone species, Microcoles vaginatus to Microcoles trintripiae. I calculated that there might be 15 million tons of cytoamine around in areas that are populated by biocrest that are warming terrestrial soils. And this parameter of microbial control of albedo was not taken into account into climate prediction, while albedo is a very important parameter. So that's why this study was important, and also kind of linked directly microbial diversity and microbial processes to soil emergent property that can impact our understanding of soil more globally. So moving on with what I want to do in my lab. So I'm going to keep working on soil microbiome. So integrating those big questions about climate change, food production, and land restoration that I presented at the beginning, what I really want to do is to try to work in the context of the soil. So I already told you, that the soil, like this biocrust, is a complex mixture of organic molecules, minerals, but also a soil atmosphere and a soil fluid, which is not only water, it's like a very complex mix, plus all the cells and all the porosity. So I think it's very important to start, addressing the question of microbial functioning within this complexity, even though it's pretty hard. So one of the first questions that I will ask is, who are the active microbes in soil? Probing active microbe, as you saw, is a good way to try to link them better to the soil activity. And then how will they answer climate change? I also will want to develop some imagery to look at where are the cells in soil, and if in fact they have the ability to talk to each other, which is the assumption that we all make when we do omics and we do correlation. In soil it's not always that straightforward, because even if two OTU correlate, like their abundance correlate in time, it doesn't really mean for sure that they talk to each other through the exchange of metabolite, for instance, because they could be in different porosity. And then one last question that I'm interested in to follow up, probably through collaboration, is how can we manipulate this community, and if there is way that we can do it in a predictable way. Finally, I also want my lab to be kind of broadcasting this idea that soil microbiome are very important through teaching and outreach, and how they serve services for communities. So I have some ideas, but I'm also happy to discuss how we could do that. And about potential collaboration, so Sharifa mentioned already that we are going to start working together, and I have also other things that I'm starting to discuss with other PI in the group. So I'm very happy about that. But like for me, I would like to work more on plant microbe interaction, knowing that I'm not really on the plant side, and soil agriculture. So I know this is. I also would like to establish a program locally. Here, so I'm looking forward to having people that know some of the soil around here, so we can develop some project here. I'm also interested in working more with viruses. If some people are interested with that, I think there is a very clear link between microbe activity, microbial activity, and how their activity is it changes with time with the dynamic of virus population. I'm also interested in developing methods. So maybe some people from other fields also look at, what kind of imagery or what kind of new sequencing stuff we can do in microbiome. And finally, I'm going to keep working on cyanobacteria and on their environmental role. So that could also be a potential collaboration area. And with that, I would like to thank my former advisors from the place I worked with. So I cannot really list everyone I've been working with. So those are just the main group leaders. And those are the reference for the two studies that I kind of developed a little bit more in this presentation. And thank you for your time. And I am happy to take any question.
Thank you, Estelle, for this amazing presentation. Very intensive and very limited time. Thank you so much. So I think we over time now, but we can probably take a couple of questions very quickly. Anyone, guys?
I'm curious to know if the Bondcat method would work with phyloplane bacteria.
With what?
Phyloplane bacteria living on leaf surfaces, or the.
Oh yeah, phylosphere, like yeah. Well, yes. I mean, why not? So in fact, there are some studies that looked at that applied it to plant leaf by immersing the plant with in the Bondcat solution for 2 minutes. So the leaf themselves, you know, and they looked at the leaf protein themselves, the plant tissue, but this also would label the phylosphere bacteria for sure. And so just so you know, like as of now, we didn't find any limit as to which taxa can take that up. So it labels all type of candidate phyla from bacteria and archaea, and it labels fungi and it labels plant, and so potentially yes.